Team 19, and we'll be talking about the use of peptide binding in shaping the T-cell, B-cell collaborative response to HIV infection. This work was done in 2013 by Dr. Samuel Landry and his team at Tulane University. For a little background, there are two main types of immunity, innate and acquired. Innate immunity is a nonspecific first line of defense, while T cells and B cells are involved in adaptive immunity, which targets specific foreign invaders or antigens that the body has already encountered. B cells attack antigens free floating in body fluids like blood or lymph, while T cells attack infected cells. HIV harnesses CD4 T cells for its own viral replication through the binding of the GP120 glycoprotein subunit to the CD4 receptor, leading to endocytosis and membrane breakdown. HIV reverse transcriptase converts single-stranded viral RNA into double-stranded DNA, which enters the nucleus and integrates into the host's genome. Normal mitosis occurs, which includes viral transcription. mRNA translation results in HIV structural proteins, and viral RNA molecules are packaged and exocytosed by the cell to infect other T cells.
From previous studies, it is believed that CD4 plus T cell works together with B cells and the immune system. CD4 plus T cell increases the antibody affinity to antigen. The B cell antigen receptor can affect T cell epitope presentation by either inhibiting or favoring pathways for antigen processing. However, relationships of epitope-specific T cell and B cell responses within a single antigen have not been reported. Understanding the mechanism of how B cells are activated and the interaction with CD4 plus T cells will tell us immunization strategies, such as antibodies binding to epitopes, in this case, CD4 binding site and B2 loop on the virus. CD4 plus T cell itself may be hard to fight back HIV GP120 protein, but might work with collaboration of B cell.
Mice that were 6 to 8 weeks old were intranasally injected with one of 3 different primers, unprimed control and GP120 peptides. Treatment peptides are peptide 2 and peptide 29, found at the N terminus of GP120 and V3 loop, respectively. Some mice received biweekly treatment of GP120 variant and a heat labile toxin, and then were sacrificed a week after last boost. Splenocytes were harvested from mice and added to cell culture bead plates used for cell isolation. Splenocytes were washed with solution, and plates were analyzed using Bioplex 200 to obtain standard curves. An ELISA plot assay was used to determine levels of cellular immune response. A CLT immune response analyzer was used to analyze the ELISA plot.
The graph on the right shows correlation of TB responses in unprimed and control-primed mice. Each treatment group showed clear correlation between T cells and B cells, and these were statistically significant. So what does this show us about antibody specificity? It shows that it's directly correlated with the specific T cell response. So an antigen might elicit a few different T cell responses together, but each T cell response in itself recruits its specific antibody response. This graph on the right shows mouse samples that were primed with either peptide 2 or peptide 29. In general, peptide priming enhanced T cell responses significantly and also impacted the antibody responses. So in conclusion, T cells play an integral role in determining antibody specificity. Injecting peptides found in GP120 helped improve the amount of T cell responses found in the cell, which also led to an increase in antibody production. Why is this so? With peptide priming, there is a B cell dominance where the B cells already know which T cells to recruit for when there is a foreign antigen. In this case, it is the GP120 glycoprotein found in HIV cells.